Okay, so welcome to this next video. So what we're doing is we're discussing the mechanism of action of the drug methotrexate. Okay, so what we have decided is that methotrexate, by inhibiting the dihydrofolate reductase enzyme, it's going to inhibit the production of tetrahydrofolate and thereby the production of the N5-N10 methylene tetrahydrofolate, which is required for the action of thymidylate synthetase. So if thymidylate synthetase cannot work, then you can't produce any thymidine monophosphate. And we'll talk about what the consequences that is going to have for the cell are, the consequences of that are for the cell in a moment, and how that's going to stop cancer cells from dividing. Okay, but for now I just want to show you the structure of methotrexate first, so that we can understand how it actually achieves its function, because it's almost identical structure to folic acid. Okay, right, so we start off with this pyrimidine ring down here, and I've totally forgotten to put the nitrogens in. So here is a nitrogen, and here is a nitrogen. Okay, and this is actually a pyrimidine ring. This actually does have alternating double and single bonds. You have a double bond there, double bond here, double bond here, and instead of the carbonyl group up here, which you would have in folic acid, you just have an amino group. Then you have another amino group here, which is the same as in folic acid. You then have this pyrazine ring here, which has the two nitrogens opposite one another, and it's still intact at this stage because this is a folic acid analogue, not a dihydrofolate or a tetrahydrofolate folate analogue. Okay, you then have this methylene group, and then the nitrogen coming up here. And now the next modification is that of this nitrogen, instead of having a hydrogen as you would have in folic acid, you have a methyl group, and that's the second alteration that occurs in methotrexate. Okay, and then you have the benzene ring, and after this it's all the same. So benzene ring here, and then you have this carboxylic acid group, which is involved in this amide link to the glutamic acid molecule. Okay, so here's our alpha carbon, here's our carboxylic acid group coming off up here, and then here's our R group here, with the carboxylic acid group right at the end. Okay, so this is the structure of the drug methotrexate. So basically it's a competitive inhibitor of the enzyme dihydrofolate reductase. So it goes into the active site, looks exactly like folic acid, but can't undergo the same reactions that folic acid can. So it just sits in the enzyme and inhibits it basically, and stops it then converting folic acid into dihydrofolate, and then dihydrofolate into tetrahydrofolate. Okay, so let's now discuss the consequences of this for a cancer cell. So why do you need thymidine monophosphate? Well, basically, you need this in order to synthesize DNA, because DNA is built out of thymidine monophosphate molecules. It's not actually used directly in DNA. Instead, what you have to do is add two more phosphate groups onto it. So you have to convert it into thymidine triphosphate before you can actually add it onto DNA. But the point is that you get the thing that you add on to DNA from thymidine monophosphate. So if you can't produce thymidine monophosphate, then you're not going to be able to synthesize DNA, because this is an essential part of DNA. So let me just give a little bit of a talk on the structure of DNA. Okay, so remember just a little bit of basic fact. DNA is basically made out of these nucleotides, which have a phosphate group, and now I'll just draw them as cartoons rather than actually skeletal structures, which have these phosphate groups, and then a deoxyribose sugar here, specifically 2-deoxyribose as we've seen already, and then it will have an organic base here, now demoted to just being a black box. Okay, and this organic base will have some, well, it'll be one of the four basically, so it can be adenine, thymine, guanine, and cytosine. So you now see why thymidine monophosphate is utterly essential, because this is thymidine monophosphate here. This is exactly this structure we drew here, just in cartoon form. And this will then be bound to another nucleotide below it. So the third carbon will have an alcohol group off, and this will be linked via a phosphoester link to the phosphate group of the nucleotide below it. So here comes the next nucleotide. So there's the phosphate group, here's the deoxyribose sugar, and then it will have an organic base here. So let's say this is guanine. Okay, and then what you'll also have is the complementary strand in DNA. Okay, so this will have the complementary organic bases. So the complementary organic base to thymine is adenine, and those two are linked by 2 hydrogen bonds. Okay, and then the complementary organic base to guanine is cytosine, and those two are linked by 3 hydrogen bonds. Okay, and then the sugar phosphate backbone consisting of the deoxyribose and the phosphate groups runs anti-parallelly on this other strand, so the ribose sugar is going to be upside down relative to the ribose sugars over here. Okay, so here's this phosphate group, and now let's draw this next nucleotide. So here's the next ribose sugar with this link between the phosphate group and that one. Okay, so I've just drawn 2 organic bases bound to, well, 2 nucleotides bound to one another. So a strand consisting of length 2, but of course this can go on and on and on, and for however many base pairs, and sometimes the chromosomes are up to, 100 million base pairs, so massive great polymers of DNA. Okay, right. So when you make more DNA, you have to have this thymidine monophosphate nucleotide in here. Now, you don't actually directly use thymidine monophosphate to stick onto the DNA. So say I wanted to add on another thymidine, another thymine nucleotide, I do not use thymidine monophosphate directly. Instead, what you bring along is a thymidine triphosphate. Okay, so here comes the thymidine triphosphate. So this is the deoxyribose sugar down here. Okay, with the thymine organic base off the side here, and now 3 phosphate groups on. So this is thymidine triphosphate, or TTP. Okay, would be another name for it. Thymidine triphosphate, TTP. Okay, now when you want to actually add this onto the DNA, what will happen is you'll cleave this bond here between this phosphate group and this phosphate group, and then add this phosphate group onto the alcohol group of the 3rd carbon of the deoxyribose sugar. Okay, and then you have added on your next nucleotide. So you don't directly use thymidine monophosphate, even though it is just the thymidine monophosphate that is actually in the DNA. Okay, so you need the thymidine triphosphate so that the breaking of this bond between what's known as the alpha carbon, the 1st carbon, and the alpha phosphate, and the beta phosphate, the 2nd phosphate, and this, by the way, is the gamma phosphate, so that the breaking of this bond between the alpha phosphate and the beta phosphate can provide the energy that you need to add this alpha phosphate onto the alcohol group of the 3rd carbon of the nucleotide above it. Okay, so the message of the story is that thymidine triphosphate is synthesized from thymidine monophosphate. In fact, everything that you use to make DNA and also RNA, where you're going to also have thymine nucleotides, all of these require thymidine monophosphate. So in RNA, when you make RNA, you, instead of using thymidine monophosphate, instead of having thymine bound to a deoxyribose, you have thymine bound to a ribose sugar. But again, the thymine bound to ribose is produced from the thymidine monophosphate, basically. So it's produced from this molecule. So, the moral of the story is that to produce any sort of DNA where you have thymine organic bases in that DNA, and to produce any sort of RNA where you have the thymine organic base within that RNA, you need to produce this thymidine monophosphate. So if you don't produce this thymidine monophosphate, you cannot add thymine organic bases into DNA or RNA. Okay, so this means that you won't be able to produce DNA or RNA. Now, what does this mean for the cell? Okay, so this has gone into the cell. Okay, so we've got our cell here. It's gone into that cell, and it has meant that you have no thymidine monophosphate anymore. So that means now that you have no thymine nucleotides of any sort. So no thymine in RNA and no thymine in DNA. That you have no constituent pieces for your thymine in RNA and your thymine in DNA. Of course, if you've already got DNA and RNA which contains thymine before we add the drug, then the drug isn't going to destroy that thymine, but it's going to stop the production of any more. So, what if the cell comes to, let's say, make some protein? So if the cell wants to produce a protein, what has to happen? Well, it has to go to the DNA, it has to go to the gene, and it has to produce a piece of mRNA that's complementary to the coding strand of the gene, and then that mRNA will undergo the translation process and be turned into a protein. So here comes the protein. And so let me just colour this in. So here is the mRNA in pink here, going through the ribosome, and here in green is the protein coming out. Okay, so if you can't produce any mRNA because you have no thymine nucleotides to add into that mRNA that you're trying to produce, then you're not going to be able to produce any protein. So one result is going to be that protein synthesis is completely stopped, okay, by the methotrexate. Okay, so protein synthesis goes down. In addition, let's say our cell wants to divide its DNA, which dividing cells generally do want to do. So if our cell wants to divide, okay, here, it wants to turn into two separate cells, then it needs to copy its genome, basically. It needs to completely copy all of the DNA within the nucleus of this cell. Now, is it going to be able to do this if it's got no thymine nucleotides? The answer is no. So it's also going to stop DNA replication. So these two processes, protein synthesis and DNA replication, are absolutely essential for cells which are wanting to divide, basically. So in order to divide from one cell to two cells, you need to divide the genome, which this drug is going to stop you from being able to do, and you also need to copy all of the proteins. So, you know, all of the essential proteins for life, they need to be duplicated. You need to make double the amount, basically. Okay, because you now need to have enough protein for both of the daughter cells that you're going to produce when you divide. So, if you have blocked protein synthesis with this drug methotrexate, then it's not going to be able to produce new protein for the two daughter cells, so it's not going to be able to divide, basically. So by blocking protein synthesis and DNA replication, methotrexate is going to stop cells dividing, so it's going to lead to cell division going down, and this is why it's an incredibly effective anti-cancer chemotherapy. Okay, because it will go into cancer cells, it will stop them from producing any sort of nucleotide with thymine in it, and by doing that it will stop them from replicating their DNA and from producing proteins, and if they can't do that, they can't divide. Okay, now this is also the basis of its side effects, because if you stop all protein synthesis within cells and all DNA replication, that's going to have problems. It's not as though this drug only goes into cancer cells. That would be fantastic, but of course it goes into every cell of your body, so it will be inhibiting this dihydrofolate reductase enzyme in every cell of your body. Okay, and specifically the cells which will be hit hardest are those which actually need to divide physiologically, of which there are many examples. For instance, the cells which produce your hair. They divide very rapidly, and when you take anti-cancer chemotherapy, a famous side effect is that you lose your hair, and that's because of the effect on these cells that produce the hair. Okay, so this drug can have a number of serious side effects, and it can be very, very toxic on the normal cells of your body. However, there is another drug that we can give to try and limit the damage that the methotrexate drug does to the normal cells of the body, whilst not helping enough the cancer cells. So the cancer cells will still be hit by the methotrexate, but the normal cells will be in some way protected against its toxicity. Okay, and we'll see this next drug in the next video.